The new corona test, LampSeq, which was developed at the University Hospital Bonn, has properties that are well ahead of those previous rapid antigen tests and even qPCR tests.
The antigen rapid tests, they are fast, but they are not as sensitive as the qPCR, which is our current gold standard in diagnostics. The qPCR, however, is rather expensive and is not as scalable as we would like it to have. So that's where the LampSeq test comes in, because we can basically provide a sensitive test that is scalable at the same time.
With the help of sequencing machines, thousands of samples can be tested simultaneously without much effort. The samples can also always be traced back to each tested individual without further retesting, as necessary with CPR pool tests in schools, for example.
And that's an advantage with LampSeq, because with the molecular barcodes, we know right away which person it is, and we don't have to go back and retest the entire class. When we put the samples into the reaction plates, then we have individual barcodes for each person or for each sample. And these barcodes are 10 base pair long base pair sequences, and they are read out on the sequencer. And it's like a house number on your street, basically. And it is incorporated into the virus during amplification. So it's only added if the virus is present. And then the sequencer can read out the sequence, and we can put it back to the individual person because we know at which part of the reagent plate there was which barcode.
A special feature of LampSeq is that it requires relatively simple infrastructure. In the end, we need a very complex machine, a sequencer, and that's a high-tech machine, but other than that, we basically just need heating devices and water baths and normal laboratory infrastructure. That makes it really scalable and potentially cheap to use. For IT infrastructure, we actually use normal laptop computers because the data we're generating in the last sequencing step is relatively small. It's in the size range of megabytes. So it's easy to analyze on a standard computer. So a current sequencing machine is basically functioning like an automated microscope. What you put in is a solution of DNA molecules. Each of these single molecules gets fixed or immobilized on a glass surface and amplified. So it is amplified so that several thousand molecules with the same sequence are produced in close proximity on the glass slide, like a spot, a dot of DNA with the same sequence. And then the microscope can detect the 4 DNA bases, G, A, T, and C, by 4 colors. So it takes an image in 4 colors, does an enzymatic reaction to read the next base, and does another image. So it's back and forth between imaging and enzymatic reaction. In the end, from the sequence of colors, the machine can then tell which DNA sequence was present in every spot.
During the development of the mass test, the question of logistical implementation arose in particular. How do so many samples find their way to the lab? To find out, the researchers set up their own test center at the UKB, where employees could be tested. It turned out that the testing works extremely quickly, which means that throughput can be very high. Such a mass test could be particularly useful in large companies and schools.
When you think of schools, it's pretty the same way. You can take the rooms where the sports is normally done, and there you can have mobile stations as well. And each class takes like 10 minutes to go through the station. The swab is taken. Each student also has this QR code, and then after like 10 or 15 minutes, they are back into their classroom and can continue with school. And the advantage here compared to the currently used antigen rapid test is probably that you have the possibility to continue with the school just after 15 minutes. You don't have to repeat tests for individuals because the testing failed.
The areas of application of LampSeq go far beyond its current use during the pandemic.
We could already show that LampSeq can differentiate between wild-type SARS-CoV-2 and the Delta variant. And that's basically for free, because you're sequencing in the end. You can get the information whether you have Delta variant for free. And we imagine that we can adapt our test method to other variants that might emerge in the future relatively quickly.
Of course, we hope that this can still make a difference in the current pandemics. In particular, when we think about winter coming, it's really nice to know whether someone has influenza or has the coronavirus. And that's something where we think it can still be applied. And for the future, we envision that this differentially diagnostics possibilities are actually really helpful also for other pathogens that are around.